Hello, what an honor to follow up on your talk, Valerie. Thank you for giving me this talk a stage. I will be talking about cell-free DNA methylation quantification using immunoprecipitation coupled with digital PCR, and that enables reduced plasma inputs for colorectal cancer detection. And it's a small part, or maybe a little bit about myself. My name is Tu. I'm a fourth year PhD student, and that was part of one of my projects. Thank you. It was actually a part of a big German nationwide consortium called Outlive CRC, working together with the DKFZ from Heidelberg and UKSH from Lübeck. So I don't need to introduce you to liquid biopsy too much. It has really good advantages for being minimally invasive, and for longitudinal monitoring it's quite valuable. But the sensitivity remains limited by the low physical abundance of the cell-free DNA or circulating tumor DNA. Obviously, you can just increase the blood plasma. I mean, going back to the good old times of bloodletting, but you can ask the patient if it's still minimally invasive, probably not so good. And in some cases, it's not only impractical, but also really not feasible. Let's talk about the really strict guidelines for pediatrics or for retrospective studies where you have biobank samples, which are pretty scarce. And here, methylated DNA is quite an attractive marker, since there's enough data showing that it's quite a stable marker in circulation, since it's more packed to the proteins and more protected from the DNases, makes sense. But the challenge still remains that methylation detection still often requires up to 60 milliliters of plasma to reach relevant sensitivities. And that's what we wanted to work on. And to understand this problem a little bit more, I want to revisit the methylation analysis techniques, which are obviously the gold standard this year, the bisulfite conversion. I know a lot of people bash on it. I mean, it has really good advantages of offering base pair resolution. And the limitation here is obviously that it highly degrades the DNA up to 90%. And what I wanted to focus on was the method of affinity enrichment using the cell-free methylated DNA immunoprecipitation protocol, or short cfMeDIP. It does not offer base pair resolution for the methylation calling, but it preserves the DNA integrity. And we hypothesized that you actually don't need the base pair resolution to distinguish cancer from non-cancer. And also the hypothesis was, since it's preserved the DNA, that with a cfMeDIP protocol, we can enable reduced plasma input while maintaining relevant sensitivity. Now I want to make a very short motivation for the model that we use, CRC in that case. And for me it was quite striking. It's the second leading cancer-related cause of death. And every year it claims 900,000 deaths. And I checked the numbers to get the head around the number. It coincides with the number of population in Amsterdam, or 3 times of Ghent, I learned this morning. And the interesting thing is really that the incidences of patients at age 50 or older is stabilizing in Western countries, also thanks to good diagnosis and therapies. But there's a striking increase in incidences of people younger than 50 years. So that is quite remarkable, and it really motivated me to eat less instant food, instant noodles, and also motivating for that project, right? So the goals were the following: to leverage first the established methylation markers for CRC detection. There are already great markers out there, and we wanted to kind of lower the barriers for clinical adaptation. And we want to integrate the cfMeDIP protocol with digital PCR for the quantitative readout and reduce the plasma input to only 2 milliliters of plasma per assay. And we want to achieve CMS performance targets. Okay, now I understand. You don't talk too much into the microphone, then it sounds better. Do you hear me still? That's much better, actually. Okay. So we wanted to achieve the CMS guidelines. They are from the US, from the Medicaid and Medicare thresholding for assays to be reimbursed in the US. And they say that the assays need to be higher in sensitivity of 74% and at a specificity of 90%. So the protocol goes as follows. We start with extracting the cell-free DNA from 2 milliliters of blood plasma, and we use 53 microliter of that eluate to put into the cfMeDIP protocol. And prior to that, we top it up with lambda filler DNA to 100 nanograms to normalize it for the cfMeDIP protocol. Then we divide it into 2 fractions. One fraction stays as the input control, which is 10% of the sample we keep, and that should represent the total fraction, let's say, the methylated portion and the non-methylated portion. Then 90% of it gets subjected to the antibody binding. They get pulled down, washed, and then eluted. And then at the end, we perform digital PCR for both the IC and IP sample, and then we can calculate the methylation ratio, which is our diagnostic metric in this case. Now, we started with a panel marker selection, and we chose these 2 publications and got these 3 markers from them, KCNQ5, C9orf50, and CEP9. And these 2 publications validated their markers using, first of all, bisulfite-based methods, using a lot of plasma volume, up to 60 milliliters, and they could validate it on plasma samples already. So they were really, in our sense, trustworthy markers, and we wanted to leverage that. Next on, we redesigned the prime and probe assays since they were based on bisulfite-based methods, right? They were targeting converted sequences, and in the cfMeDIP protocol, we need to target the native sequences. And we used multiplexing by employing the population-specific reporter system from our in-house technology. I don't want to go too much into detail there. We use mediator probe and the population-specific reporter, but I highly advise you to visit poster number 4 and 5 to get more information about that. Next up, we did a technical validation first with spiken material. We used cell culture material. So for the CRC model, we used HCT116 cell culture, extracted mononucleosomal DNA from it to represent the cell-free DNA fragmentation patterns. And you can see that for all 3 markers, we could go down to 1 nanograms of input and got methylation ratios which were detectable still. And when inputting 15 nanograms of wild-type cell-free DNA, the methylation ratio stayed below the threshold. And on the left-hand side, you see a representative scatter plot of the IP and IC sample, as well as NTC. And I still have a lot of time, so maybe I tell a little bit of an anecdote here. I didn't know that there are qPCR fans here, otherwise I would have included qPCR data, because I'm actually a big qPCR fan. I worked in a qPCR company before, and then my supervisor actually told me to go directly to the digital PCR, but I told him, no, let me try qPCR first. I did that. It didn't work so well. You should have seen his face, though. He was kind enough to not say anything, but I put in 100 nanograms of cell culture input inside there, and the CQ values were above 30. Then I didn't even try to go lower. And 100 nanograms of tumor DNA in your plasma sample is not viable, probably, for the patient. So, but with digital PCR, it worked. So I converted as well. Closing up with a clinical validation, we used a cohort of 32 early-onset CRC patients and 29 age and gender-matched non-CRC controls. And the median methylation ratio for each marker was significantly higher in early-onset CRCs. Then we did a receiver operating characteristics curve, then set the threshold to have the specificity at 90% according to the guidelines, right? And extracted those sensitivities, which you can see here in this plot. We also included a very simple algorithm of considering the individual as a CRC patient if at least one of the markers gets above the threshold or at least 2 of them. And you can already see that the single marker of KCNQ5 is already the best performing one with 85% sensitivity at 90% specificity. And when we deconvolute it a little bit more, you can see that they actually significantly correlate with the tumor stages, and obviously it has the highest sensitivity with stages 3 and 4. Bless you. So let's revisit the goals. First of all, we managed the 3-plex assay. Then we used the methylation ratio as a diagnostic indicator. We could reduce it 8 times less than the sensitivity matched bisulfite assay. When you actually calculate it down, how much we use in the actual dPCR, we can even say that we only use 500 microliter of the initial plasma sample, so 20 times more, 20 times less than the sensitivity matched bisulfite assay. And we could validate it within a cohort with a mean age of 43 years. And we had a sensitivity of 85%, as I said, at a set specificity of 90%. So last slide here, I want to give a very exciting outlook, which we are still working on. We want to expand the 3-plex assay by 2 additionally therapeutically relevant biomarkers, which were already mentioned yesterday from Johan, MGMT and MLH1. And also the resulting data we want to include in a multi-analyte panel. So that was actually also the goal, to reduce the plasma volume. So we only need to dedicate 2 milliliters to the methylation assay, and the rest of the plasma can get tested for mutations or other markers. So really excited about the results there. And let's see if it also increases their sensitivity if we expand the triplex to the 5-plex, which is not always the case, I guess. And I want to give a big thank you to my 3 colleagues here, Clara, Peter, and Judith. I started the project with Clara and Peter. Peter is my supervisor, so he was the guy who recommended me to use digital PCR, not qPCR. Clara did a lot of troubleshooting to get the triplex assay working, and Judith is the one who's working on the Outlook goals for us. And I also want to thank the funding buddies and for you for your attention. And I'm ready for the questions.